So welcome everyone to my presentation. I would like to start by thanking Francesca, Felix, and Ishir for giving me this opportunity and actually encouraging me to apply for a talk in this fantastic seminar series that I've been with from the very beginning. So today I wanted to tell you about my work from my time at Reinhard Jan's lab. And as Francesca said, in the meantime, I already moved to Berlin to work with Volker Hauke. So one of the most well-studied membrane fusion events in cells is happening during synaptic neurotransmissions when synaptic vesicles fuse with presynaptic plasma membrane in response to electrical stimulation. And this fusion process is mediated by proteins belonging to family of SNER proteins that mediate membrane fusion along the trafficking pathway in all eukaryotic cells. In the case of synaptic vesicle fusion, we have 3 of them involved. Syntaxin and SNAP25 are located on the presynaptic plasma membrane, and synaptobrevin is on synaptic vesicles. And when these 3 proteins assemble into so-called SNER complex, they zipper and bring the membranes closely together, and by this they induce membrane fusion. And looking from a more physical or membrane-centric perspectives, membranes, in order to fuse, need to go through a number of intermediate steps, and these intermediate steps are determined by the energy barriers along the fusion pathway. So first, membranes need to come closer towards each other, then they need to come into direct physical contact to finally form a fusion stalk where first lipids place tails in between the 2 membranes. This then transits to either hemifusion diaphragm or to formation of a fusion pore. And today during my talk, I will concentrate mainly on these very early stages before the hemifusion has yet occurred. So from previous work, we know that if we have some mutations within the SNER motif, we can stop the fusion reaction at certain stages. So for example, here we can see that this reaction was stopped at the tightly docked intermediate. If we then have mutations closer to the plasma membrane or closer to the membrane, we have the reaction inhibited at the hemifused state. In particular, there is one interesting mutant of synaptobrevin with single amino acid deletion as position 84 that leads to accumulation of these tight intermediates where membranes appear to be in physical contact, but we don't fully understand yet. Another interesting mutant of synaptobrevin is a double alanine substitution located further away from the membrane. And this mutant was shown before by the labs of Dirk Fasshauer and also Jakob Sorensen to slower SNER complex assembly and also inhibit exocytosis in chromaffin cells. So further studies of docking of liposomes and the resistance of liposomes containing the synaptobrevin mutant to SNER complex disassembly showed that if we put the disassembly machinery NSF-alpha sub, we can undock the liposomes docked by the SNER complexes. However, over time, there's accumulation of vesicles that would not go away from each other. So we have this very stable intermediate that becomes SNER independent. We then also quantified the distances between membranes of docked vesicles, and it turned out that we have indeed 2 populations of these docked vesicles, one with a peak around 0.1 nanometers when membranes actually appear to be in physical contact, and with second one where there's a little bit distance between the membranes where potentially SNER complex could fit in. So with this, we have 2 different docking stages that are different by the SNER tethering dependence and also membrane-membrane distance. So in order to further investigate these 2 different docking stages, we wanted to understand the dynamics of vesicles docked on the membrane. And in order to do this, we used the experimental system with giant euromellar vesicles that had plasma membrane SNERs reconstituted and smaller liposomes with synaptobrevin. And then in order to perform 3-dimensional tracking of these docked liposomes, we used interferometric scattering microscopy that allowed label-free imaging. So here you can see example of the raw data that you get from this kind of microscope. These big rings, they correspond to the scatter signal coming from the GUV itself. Here, this is the bottom part of the GUV. And the smaller points that move in the movie, I hope you can see that, these are liposomes that are docking, that are docked and are diffusing on the surface of the GUV. So from these movies, we were able to further extract these 3-dimensional trajectories And from this, we calculated the diffusion coefficients of vesicles containing various synaptobrevin variants. So what we found here was that together with the fusion progress reaction, we have the diffusion coefficient would slow down. So if we have the liposomes containing the double alanine mutant that leads to accumulation of the loosely docked intermediates, we have the fastest diffusion coefficients. Then for the one that leads to accumulation of tight docking intermediates, we have average diffusion coefficients. And in case of the wild type protein that normally induces fusion, but when it inhibits or stops the reaction at a certain step, then it can be also hemifusion. We notice this hemifused intermediates. So far what I have shown you is that for loose docking, these are snare dependent. So if you disassemble the snares, the two membranes would go away from each other. There is some distance in between the membranes that could potentially fit proteins. We also have faster diffusion of docked vesicles than for the subsequent fusion stages, and we think they are bridged by trans-snare complexes. On the other hand, for the tight docking, they seem to be snare independent. So they need the snares in order to first come together, but then you can disassemble the snares, remove them, and the membranes would still stay together. There is no space in between the membranes. They seem to be in physical contact as visible in EM images. And we think these interfaces are free of proteins. And on average, there is slower diffusion of such tightly docked vesicles than loosely docked vesicles, but also it seems to be faster than for the hemifused ones. But we were still curious, what is exactly the docking interface morphology, especially in this tight docking intermediate, and how the membrane properties would change, since it seems that it's not really possible to form such intermediate with very high electrostatic repulsion going on between the head groups coming from both membranes. So in order to closer look at this tight docking interface, we designed another experimental setup with two sets of GUVs. So in one GUV, you will have plasma membrane snares reconstituted together with a membrane dye. And in the other set of GUVs, we had a synaptobrevin docking mutant that was labeled with a fluorescent dye in order to monitor the position of both membrane and also proteins in our system. So if our hypotheses are correct, that in case of loose docking, we have this bridging by trans-snare complexes over time, we should observe in the system accumulation of snare proteins at the interface because we don't have any recycling machinery. So we would see enrichment of green fluorescence at the interface. In case of tight docking, on the other hand, we should have protein-free interfaces, and therefore the protein signal would be not present at the interface. And indeed, this is what we were able to observe in our GUV system. So in some cases, we had this increased fluorescence at the membrane interface, and in other cases, we had the protein signal removed from the interface. At the same time, we were sure that this intermediate is not hemifusion because otherwise the lipid dye would diffuse into the membrane of the green vesicle. Furthermore, we could also demonstrate that these loose and tight docking intermediates are actually productive fusion intermediates. They are not on the off pathway. So here we start by the loosely docked vesicle pair. So we see the enrichment of green fluorescence at the interface. And then after a while, we see that protein is removed from the interface and there is diffusion of the membrane dye into the green vesicle. So the hemifusion has occurred. And finally, after a while, both vesicles fuse together and quickly relax into one single vesicle. We could also observe fusion happening in the tight docking. So here you can see a tight docking vesicle pair with no signal coming from the protein at the interface. And then we, in this case, it directly transited to fusion. So you see that two vesicles are fusing and then relaxing quickly into single vesicle where both protein and membrane signal are mixed. So we were further curious how would electrostatics influence formation of these loose and tight docking interfaces. It is known for a while now that calcium can simulate fusion of phosphatidylserine containing vesicles. So I looked at the formation or fraction of vesicles that would be loosely or tightly docked in the presence of various concentrations of calcium. And one finding was that we basically could observe tight docking intermediate even without any divalent cations present in the buffer. But with increasing concentration of calcium, we had more and more of the tight docking being formed, suggesting that calcium could modulate formation of the tight docking. We also approached the electrostatic question from the membrane perspective, and in one of the membranes, we incorporated 1% of Pi45P2. And in this case, we saw substantially less of the tight docking interfaces being formed, again suggesting that higher electrostatic repulsion coming from the membrane would inhibit formation of the tight docking interface. So finally, I also looked at the morphology of the membranes in cryo-EM images. And what I found was that at the tight docking interfaces, there is substantial thickening that can be detected. So here you can see that for the tight docking interfaces, their membranes become much thicker, and the same membrane, but outside of the docking interface is actually has normal thickness as for the free vesicles, for example. And this effect is much bigger in case of tight docking in comparison to loose docking. And in order to further investigate this very interesting effect of membrane thickening, we teamed up with the group of Helmut Grubmuller and Leonard Heinz, who is, I think, also today with us, performed atomistic molecular dynamics simulations of two membranes approaching each other. So in these simulations, we kept the same lipid and buffer composition as in my experiments. We just didn't have proteins. And to our delight, with this system, we were able to reproduce this membrane thickening together with closer distance between the both membranes. At the same time, we could monitor many more parameters. So we also determined that there is decreased membrane area and also decreased membrane volume together with decreasing distance between two membranes. Then Leonard also took a closer look at other structural changes happening in the membrane with decreasing distance between both membranes. And what he found was that in the inner leaflet, so in the leaflet that is directly facing the other membrane, lipid head groups are tilting their head groups, basically. So what we hypothesized from that perhaps there is, due to dehydration that is going on at the very close distances, then the electrostatic environment of the membrane would change, and this would induce this compensatory effect of head group tilting. So finally, also with molecular dynamic simulations, we could determine the order of structural rearrangements in the tight docking formation. So here Leonard performed time response analysis. So he found out that head group tilt is the first event that happens, and then this is followed by reduction in membrane area. And this is finally followed by increase in membrane thickness. So now we would like to put our findings into a broader context of energy landscape of membrane fusion. So here, this is a very naive way without taking into account height of the energy barriers. But basically, snare assembly would lead to formation of a loose docking intermediate that is located in the first energetic valley. And this is then facing the energy barrier that is basically generation of protein-free membrane patch and dehydration, as well as overcoming of electrostatic repulsion between two approaching membranes. And when this is overcome, then we have formation of this tight docking intermediate that also represents energetic valley. And then the next step is when the first lipids place its tails in between two membranes, and we have formation of a stalk and then a hemifusion intermediate. If we then take a closer look at formation of the tight docking interface, we think that first step is then dehydration due to fact that there's less volume in between the membranes, water is removed from this space. Then this leads to some electrostatic rearrangements in the membrane that are compensated by increase in head group tilt. And this is then followed by shrinking of the area of the membrane as a response to this head group tilt. And this finally squeezes the tails of the lipids, and they react by straightening out and increase in membrane thickness. So with this, I would like to convince you that tight docking intermediate is actually physiologically relevant. So it can be noticed, for example, when synaptic vesicles are docked at the presynaptic plasma membrane. We think that these images represent a state where the hydration barrier has already been overcome, and perhaps it can generate vesicles even with higher release probability. Tight docking can be also observed in membrane fusion mediated by viral proteins, and also in fusion of mitochondria. So with this, I would like to acknowledge people involved in this work. First of all, Reinhard, for giving me the opportunity of performing this project in his lab. The IceCut microscopy was done in the lab of Vahid Sandogar in Max Planck Institute for the Science of Light. And molecular dynamics simulations were performed in the group of Helmut Gruppmüller by Leonard Heinz. I would also like to acknowledge Volker, who is supporting me since I moved to Berlin. And again, thank you for giving me the opportunity of sharing this story here. I'm looking forward to the questions.